Hello everyone. Yeah, this is my talk. GPCRs represent the largest family of membrane proteins targeted by FDA-approved drugs. They control cellular responses to external signals, regulating neurotransmission, immune responses, and more. Here's the challenge. We have approximately 800 GPCRs in the human genome, yet around 80 remain orphaned, so that means they have no natural ligands. Our focus is chemokine GPCRs, which play central roles in inflammation, immunity, and cancer, but we have surprisingly few approved drugs. Each orphan receptor represents enormous untapped therapeutic potential. Previously, by deorphanizing GPCR25, we've seen how traditional manual approaches have proven successful but highlight a scalability problem. Here we use single cell expression, tissue expression, conservation, binding scores, sequence motifs, isoelectric point, molecular weight, and other similar features, as well as docking methods with structural similarity scores. But we did each of these step by step. We used significant resources and time investments to complete this computational process, so we wanted to scale this up to address the dozens or all of the remaining orphan receptors. This motivated our computational approach. Our solution transforms deorphanization from manual to systematic using an integrated AI pipeline. We combine machine learning-based genome scale ligand scanning with AlphaFold2 multimorph structural modeling for atomic level binding insights. Multi-omics integration incorporates transcriptomics, sequence motifs, and structural features, as well as all the previous features we've described earlier, to create comprehensive biological context. Our goal is discovering novel druggable pairings for both orphaned and characterized GPCRs, dramatically reducing the time and cost versus traditional approaches such as experimental or the previous step-by-step approach, while enabling systematic exploration of the entire GPCR ligand interaction space. So here, structure provides residue level insights into binding specificity and selectivity, so protein structure is really important. Our alpha pull down plus AlphaFold2 multimer implementation uses pre-calculated MSAs and generates models in seconds through HPC parallelization. We've created approximately 2 million structural models of a systematically generated dataset of validated and new or unknown GPCR ligand complexes. This enables large-scale screening and provides the structural foundation for machine learning analysis. Each model captures potential binding geometries and interaction patterns that would be impossible to explore experimentally. For feature engineering, we specifically wanted to translate biological information into computational features for input. For biological context, we analyzed tissue-specific expression or co-expression using RNA-seq data from HPA and GTEx datasets to identify where receptors and ligands naturally co-occur and create correlation features. We incorporate sequence motifs, so the location of DRY and other important motifs in GPCRs, biochemical properties, and coevolution scores derived from blast analyses, capturing evolutionary relationships. Structurally, we extract residue-level contacts using Ballesteros Weinstein numbering for consistent alignments, AlphaFold confidence metrics, and distance measurements between ligand termini and a receptor domain. This allows us to ask whether they bind, but also whether it's biologically plausible. This is an overview of our preliminary ML model. So here is our architecture. We use transformer-based architecture for binary interaction prediction. So it uses input projection, expanding features to 256 dimensions, sinusoidal positional encoding for sequence information, and a 4-layer transformer encoder with 8-head self-attention, capturing diverse interaction patterns. The multi-head self-attention is key here. It means each residue can look at and attend to every other residue in the binding, or in the structure, and learning which residue pairs are most informative for binding prediction. This also allows us to focus on different types of residue relationships. We also use comprehensive regularization, including dropout and weight decay. Then we concatenate the additional features which have been batch normalized, and then we run it through a multi-layer perceptron with 4 layers. We also use cross-validation to ensure we capture variability. And a major challenge that we had was data set imbalance, as we had much less validated interactions versus many potential negatives. So we required random sampling to create balanced data sets. These are our initial results. We achieved an AUC of 94.67% and around 88.7% accuracy across 5-fold cross-validation, which is promising given the problem difficulty and limited training data. More importantly, we identified several high-confidence novel GPCR ligand pairings that are currently undergoing experimental validation through functional assays and binding studies. We also are able to compare the importance of these features, and early results suggest that biological features are important as well as the physical structural interactions. Most importantly, a key advantage of our model is how interpretable it is, and our eight-head attention method reveal which residues drive each prediction, providing direct experimental validation targets. Average attention scores correlate with well-known functional domains, enabling specific mutagenesis hypotheses. So yeah, as a conclusion, we were able to transform GPCR deorphanization from like slow and like the traditional method to very fast, high throughput, systematic methodology. And next steps include scaling to all orphan class A and B GPCRs. So this would include running like the entire family across all of the predicted ligands that we've also been working on. So we have a separate kind of like project where we're trying to predict potential peptide ligands. So we're also going to finalize the transform architecture and fine-tune it to increase the accuracy. And overall, this pipeline represents a new paradigm for ligand discovery applicable beyond orphan receptors. So we could revolutionize early stage drug discovery. Yeah. For acknowledgements, this would not be possible without the help of my mentors. So Kevin, my PI, Dr. Butcher, and also our collaborators, Sophia and Dr. Kufarina Revna.
Okay, thank you very much. Thank you very much, Catherine. Because we're kind of running short of time behind a little bit, we will forego question now, but sure,